सो कायस्माटा फॉर्मेशन कब होती है? सो हम डिप्लोटिन में देख सकते हैं, डिप्लोटिन में is recognized by the dissolution of synaptomal complex and X-shaped structure are called chiasmata. सो वो डिप्लोटिन में फॉर्म होता है। क्रॉसिंग ओवर कब होता है? सो हम पेकाइटिन में देख सकते हैं, पेकाइटिन में क्रॉसिंग ओवर इज द एक्सचेंज ऑफ जेनेटिक मटेरियल, वो पेकाइटिन में होता है। नेक्स्ट, सिनेप्टो सिनेप्टोनेमल कॉम्प्लेक्स फॉर्मेशन, सो वो है जो जाइगोटिन में फॉर्म होता है। फॉर्मेशन ऑफ कॉम्प्लेक्स स्ट्रक्चर कॉल्ड एज सिनेप्टोनेमल कॉम्प्लेक्स, जाइगोटिन में होता है। टर्मिनेशन ऑफ कायस्माटा डायकाइनेसिस में होता है, यहाँ देख सकते हैं टर्मिनेशन ऑफ कायस्माटा। नेक्स्ट, मैच लिस्ट 1 विथ लिस्ट 2। सो सेल्स आर मेटाबॉलिकली एक्टिव एंड प्रोलिफरेट। सो हम जीवन में देख सकते हैं, ये जीवन फेज में in during G1 phase the cells is metabolically active and continuously grows. So cells are metabolically active and proliferate in G1 phase. DNA replication take place in, so हम S phase में देख सकते हैं DNA replication होता है। प्रोटीन आर synthesized in, so हम देखते हैं G2 में phase protein are synthesized, so G2। And quiescent stage with metabolically active cells, so quiescent stage में G0 होता है, G0 phase। Next question, spindle fibers attached to kinetochores of chromosome during, spindle fibers are attached to kinetochores of chromosome at metaphase, हम देखते हैं एनसीईआरटी में। Next question, given below are two statement which are correct. So statement 1, chromosome become gradually visible under a light microscope during leptotin stage, it is correct. Next, the beginning of beta protein stage is recognized by a dissolution of synaptomal complex. So हम देख सकते हैं in diplotin stage is characterized recognized by the dissolution of the synaptomal complex. So statement 2 is correct. So both are correct. Next, following are the stages of cycle. Choose the correct sequence of stages from the option. So पहले G1 phase होता है, G1. Next, synthesis होते हैं. Next, synthesis phase. Next, G2 phase. Next, karyokinesis. Karyokinesis होती है. Karyokinesis. Next, cytokinesis. हम यह देख सकते हैं, ये पहले G1, next S, G2, and next karyokinesis and next cytokinesis. Next, match list 1 with list 2. So in diakinesis, completion of termination of chiasmata होता है. In पेकाइटिन में देख सकते हैं appearance of recombination nodules. In zygotin synaptonemal complex फॉर्म होता है. Next, leptotin. In leptotin, chromosome looks like threads. Next question. The dissolution of synaptomenal complex occurs during. So in diplotin is recognized by dissolution of the synaptonemal complex. So diplotin our answer. Next. Doubling of the number of chromosome can be achieved by disrupting mitotic cell division soon after metaphase. Next. During which stage of mitosis and meiosis respectively does the centromere of each chromosome split? स्प्लिट हम एनाफेज में देख सकते हैं, यहाँ स्प्लिट हुए क्रोमोसोम, यहाँ स्प्लिट हुए। सो इन एनाफेज पे anaphase is mitosis and anaphase 2 in meiosis. Next, the process of appearance of recombination nodules at which substage of prophase. So हम पेकाइटिन में देख सकते हैं, पेकाइटिन पेकाइटिन this stage is characterized by the appearance of recombination nodule. So our answer is पेकाइटिन. Next question, मैच लिस्ट 1 with लिस्ट 2. जो एम फेज है उसमें इक्वेशनल डिवीजन होती है, और G2 फेज है, या हम देख सकते हैं ड्यूरिंग द G2 फेज प्रोटीन आर सिंथेसाइज. सो 1, जो क्वेश्चन स्टेज है वो इनएक्टिव फेज है. Exit is given after phase enter an inactive stage called question stage. Question stage जो inactive stage है. And G1 phase is the interval between mitosis and initiation of DNA replication. Next question. Given below are two statements which are correct or incorrect. So statement 1, during G0 phase of cell cycle the cell is metabolically inactive. So हम देख सकते हैं G0 phase is question stage. Cells in this stage remain metabolically active. So our statement 2 is incorrect. It remains metabolically active. Next statement 2. The centromere undergoes duplication during S phase of interphase. So our answer is statement 2 is correct. Next. Which stage of meiosis can last for months or years in the oocyte of some vertebrates? Some diplotin में देख सकते हैं diplotin. The beginning of diplotin is recognized by the dissolution of the synaptomal complex. And in oocyte of some vertebrates, diplotin can last for months or years. So our answer is diplotin. Next. In meiosis, crossing over and exchange of material between homologous chromosomes catalyzed by the enzyme recombinase. Recombination होता है recombinase से. Next question. Bivalent or tetrad formation is characteristic feature observed during. So ये जो bivalent और tetrad है वो किसमें होता है? Synaptomal complex in zygote phase में होता है. So our answer is option 2. Next question. Which one of the following never occur during mitotic cell division? Pairing of homologous chromosome नहीं होता. Next question. The appearance of recombination nodules on homologous chromosome during meiosis is characteristic feature. Characterizes synaptomal complex, no. Bivalent, no. Sites at which crossing over occur? Yes, it is correct. Next. Regarding meiosis, which of the statement is incorrect? So statement 1, these are two stage in meiosis, meiosis 1 and meiosis 2, it is correct. In meiosis 2 stage होते हैं, meiosis 1 or meiosis 2. Next statement, in the S phase of meiosis, DNA replicate occur in 2. This is no. Cell division occurs in 1. Next statement, pairing of homologous chromosome and a recombination occurs in meiosis 1, it is correct. 4 haploid cells are formed at the end of meiosis, it is correct. Next. Select the incorrect statement with reference to mitosis. So statement 1, all the chromosomes lie at equatorial metaphors, it is correct. Next statement, spindle fibers attached to centromere of chromosome, it is incorrect. Spindle fiber attached to kinetochore. काइनेटोकोर not centromere. Next statement, chromosome decondensed at telophase, it is correct. Splitting of chromosome occurs at anaphase, it is correct. Next question, which of the following stages of meiosis involves division of centromere? हम anaphase में देख सकते हैं division हुई है centromere की. So our answer is anaphase 2. Next question, match list 1 with list 2. So S phase है उसमें S phase है जो उसमें DNA replication होता है. G2 phase है, in G2 phase protein are synthesized. क्वेश्चन स्टेज है जो इनएक्टिव फेज है और G1 is the interval between mitosis and initiation of DNA replication. Next, the fruit fly has 8 chromosomes in each cell during interphase of mitosis. If the number of chromosomes at G1 phase is 8, what would be the number of chromosomes after S phase? जो क्या होता है S phase में duplication of DNA होता है occur, बट जो अमाउंट इंक्रीज होता है नॉट क्रोमोसोम नंबर. नॉट क्रोमोसोम नंबर. सो इसमें सिर्फ अमाउंट इंक्रीज होगा नॉट क्रोमोसोम नंबर. सो इसमें क्रोमोसोम नंबर 8 है तो आफ्टर एस फेज भी क्रोम 8 ही होगा. नेक्स्ट, सेंट्रियोल अंडरगो डुप्लीकेशन ड्यूरिंग सम हम या एनसीईआरटी में देख सकते हैं ड्यूरिंग द एस फेज डीएनए रिप्लिकेशन बिगिंस इन द न्यूक्लियस एंड सेंट्रियोल डुप्लीकेट्स इन साइटोप्लाज्म. सो एस फेज आवर आंसर. नेक्स्ट, which stage of meiotic prophase shows termination of chiasmata? So diagnosis देख सकते हैं. In diagnosis, this is marked by termination of chiasmata. So our answer is diagnosis. Next, some dividing cells exit the cell cycle and never and enter vegetative inactive stage. This is called question stage. This process occurs after at end of G1 phase. ये देख या हम देख सकते हैं these cells do not divide further, exit G1 phase. So our answer is G1 phase. Next, identify the correct statement with regard to G1 phase of interphase. So first statement, recognition of cell component take place. No. हम यहाँ देख सकते हैं G1 phase corresponding to interval between mitosis and initiation of DNA replication. In G1 phase, metabolically active and continuously grow. So cell is metabolically active and continuously grow. Next question. In zygote में क्या होता है? साइनेप्सिस होता है. पैकाइटिन में क्या होता है? पैकाइटिन क्रॉसिंग ओवर होता है. इन डिप्लोटिन क्या होता है? इन डिप्लोटिन dissolution of synaptonemal complex. So that is called as and chiasmata form होता है. chiasmata. And diakinesis में termination होता है. termination of chiasmata. So our answer is option 1. Next, the correct sequence of phase of cell cycle. So पहले तो G1 phase होती है, उसके बाद में S phase होती है, S phase के बाद G2 and then M phase. So our answer is option 3. Next, dissolution of synaptonemal complex occurs during the final stage of meiotic prophase is diakinesis. This is marked by termination of chiasmata. And no, sorry. जो डिसोल्यूशन ऑफ सिनेप्टोनल कॉम्प्लेक्स होता है वो किसमें होता है? डिप्लोटिन में. Dissolution of synaptonemal complex. So our answer is diplotin. Next question. Match the following with respect to meiosis. तो zygote ये हमने देखा है पहले. Next question. After meiosis 1, the resultant daughter cells have twice the amount of DNA in comparison to haploid cell gamete. Next, cells in G0 phase exit the cell cycle, वो cell cycle से exit करते हैं. Next question, the stage during separation of paired homologous chromosome begins in diplotin. यहाँ हम देख सकते हैं ये the tendency of the recombined homologous chromosome of the bivalent to separate from each other. So our answer is diplotin. Next, during cell growth, DNA synthesis takes place in S phase. DNA replication begin in the nucleus. So S phase is our answer. Next, when cell has stalled DNA replication, for which checkpoint should be predominantly activated? So G1S. This is checkpoint. Next question. पेकाइटिन पेकाइटिन में क्या होता है क्रॉसिंग ओवर टेक्स प्लेस होता है एक क्रॉसिंग है पेकाइटिन में हम देख सकते हैं पेकाइटिन में क्रॉसिंग ओवर होता है मेटाफेज A1 में क्रोमोसोम अलाइन होते हैं एट इक्वेटर इक्वेटर मीन्स बीच में डायकाइनेसिस क्या होता है होता है टर्मिनेशन ऑफ कैस्मेटा एंड जाइगोटिन में पेयरिंग ऑफ होमोलोगस होता है होमोलोगस क्रोमोसोम पेयरिंग होता है नेक्स्ट क्वेश्चन स्पिंडल फाइबर attach onto kinetochore chromosome of the chromosome. Next question. Which of the following is not characteristic feature during mitosis and somatic cell synapses? Next question. In meiosis, crossing over initiated at जो पेकाइटिन है उसमें क्रॉसिंग ओवर होता है स्टार्ट होता है the site at which crossing over occurs between non-sister chromatids of the homologous chromosome. So the answer is pekitin. Next, arrange the following event. Meiosis in the correct sequence. So पहले synapses होता है synapses, then crossing over होता है, then termination of chiasmata होता है, then disappearance of nucleus होता है. So our answer is option 4. Next, during which phase of cell amount of DNA in a cell remains at 4C level if the initial amount of 2C. So only in G2 phase. Next question. In S phase of cell cycle क्या होता है? In animal cells during S phase DNA replication begins in the nucleus and central duplicates in cytoplasm. So amount of DNA is double in each cell. Next question. The enzyme recombinase is required at which stage? So पेकाइटिन में recombinase होता है. Recombination पेकाइटिन में होता है. Recombination between homologous chromosome is completed by the end of पेकाइटिन. So उसमें required है recombinase. Next question. The complex formed by pair of synapse homologous chromosome is called as जो bivalent है इसमें बोलते हैं the complex formed by pair of synapse homologous chromosome is called as bivalent. जो synapses होता है homologous chromosome का उसको bivalent कहते हैं bivalent और tetrad. So our answer is bivalent. Next. During meiosis when the chromosome start pairing at जो zygote है उसमें pairing होती है. During this they start pairing together and this process association is called synapsis. So our answer is zygote. Next. During the metaphase stage of mitosis, spindle fibers attach to kinetochore chromosome. That chromosome ने मैंने देखा है ये इक्विटोर पे लाइन है. सो ये स्टेज है मेटाफेज। नेक्स्ट, ड्यूरिंग द गैमेट फॉर्मेशन द एंजाइम रिकॉम्बिनेस पार्टिसिपेट ड्यूरिंग प्रोफेज। हमने देखा है रिकॉम्बिनेशन होता है, रिकॉम्बिनेशन प्रोफेज में। Identify the meiotic stage in which homologous chromosomes separate while the sister chromatids remain associated with the centriole. So, chromosome separate किस में होते हैं? Anaphase में. So, our answer is anaphase में. Select the correct option with respect to mitosis. So, mitosis में chromosome move to spindle equator and get aligned along the metaphase. Metaphase में हमने देखा है ऐसे इक्वेटोरियल लाइन पे लाइन होते हैं. Next, at metaphase chromosomes are attached spindle fiber by kinetochore. तो पता है हमें. Next, during mitosis endoplasmic reticulum nucleolus begins to disappear at. हम यहाँ देख सकते हैं ये disappear होने लगा है. So let prophase. Next, synapses occur between two homologous chromosomes. Next, at what stage of cell cycle are histone protein synthesized? So जो histone protein होता है DNA replication होता है, so उसमें DNA replication begins in nucleus in S phase. So during S phase. In somatic cell, DNA replication takes place in S phase. यहाँ देख सकते हैं during S phase DNA replication begins. Next question. If you are provided with the root tips of onion in your class and are asked to count the chromosome, which of the following stage you can you most conveniently locate? At metaphase. Metaphase में देख सकते हैं इक्वेटोरियल लाइन पे होते हैं और उसमें अच्छे दिखते हैं.